Welcome to Brain Ponderings. This is the fourth in a series of presentations on brain bioenergetics, neuroplasticity, and brain health. And in the first three, the first one was on just general mechanisms of cellular and systemic energy metabolism, how cells in the brain acquire nutrients and use them to generate ATP. And then the second one was on mitochondria, how they're moved around within neurons, how they can divide, and how when they become dysfunctional, they're removed. Then the third was on effects of physical exercise and intermittent fasting on neuroplasticity, health, and neural resilience. And this talk today is going to be on brain aging and neurodegenerative disorders, Alzheimer's, Parkinson's disease. It also mentions stroke um, in relation to energy metabolism. So, what seems to be happening during normal brain aging in cells that kind of compromise them and render them vulnerable to disease or injury. And then I'll finish up the presentation with some newly emerging ways to counteract specific aspects of brain aging and their roles in disease, brain disease. In this diagram, this just shows that during normal brain development, stem cells divide and then they differentiate into neurons or other cell types in the brain. In the case of neurons during adult life, the neurons can actually increase in size or number of synapses in response to things that I've talked about previously, like exercise, intellectual challenges, intermittent fasting, or in diseases, they can degenerate. And this is a quote from the Nobel laureate Francois Jacob, who says, "It's truly amazing that a complex organism formed through an extraordinary intricate process of morphogenesis should be unable to perform the much simpler task of merely maintaining what already exists." And before I move on, I just want to mention that I'm showing slides on my YouTube channel. So if you're listening on Spotify or another audio outlet, uh, and you want to or have a chance, you might be benefit from looking at the illustrations I'm presenting. Okay, so in the United States, Alzheimer's disease affects about 7 million people, and Parkinson's about 1 million, and stroke, there's about 800,000 per year and 140,000 deaths per year. In the case of AD, Alzheimer's, and Parkinson's disease, PD, there's always long-term disability, and the individuals require a lot of care. This can also be the case in stroke, where there's a lot of damage to nerve cells. And these three age-related brain disorders account for a major uh, cost to our healthcare system and also to this productivity, because not only the affected individual uh, is not productive when they're symptomatic, and they often have to have caregivers take care of them, often family members. During normal aging, our brain functional capabilities decline in everybody. And interestingly, this is already noticed in the third and fourth decades of life. And here's some data from a study done a while ago, actually, where they looked at different aspects of cognition, reasoning, processing speed, how rapid can your brain process new information, and then memory, retaining memories long term. You can see from these curves in the bottom, the average, that black line shows the average, that there's a decline in all of these cognitive domains beginning in the 30s and 40s. And I find this interesting if you look at physical abilities, you look at sports, right? Doesn't matter what the sport is, pretty much. Football, baseball, hockey, basketball. Athletes are at their peak typically in their late 20s, early 30s, and by 40, it's already clear that they're declining. Okay, so this is also true in the brain. Now, these declines, these are in normal people, so these declines are not affecting most of their ability to carry out their jobs, their occupations. I should also note from the standpoint of Nobel Prize winners, for the most part, the work for which they receive the Nobel Prize is done when they're in early stages of their career, uh, particularly when it's experiments they've done themselves and designed themselves. This is drawings from a famous artist, self-portraits when his brain was healthy, functioning optimally or at peak for him. And then he developed dementia and he did self-portraits as the dementia progressed. So within just 4 years, you see a dramatic compromise. And this is probably, this first self-portrait here in 1996 was when he was first becoming symptomatic. And so he's probably already a lot of damage to neural networks done by then. Okay, what happens in cells in the brain during aging? This diagram on the left is from a review article I wrote with a former postdoc that's on the hallmarks of brain aging. There are a lot of changes that have been documented occur in brain cells. Pretty much all of these also occur in other tissues and organs throughout the body. There's increased oxidative damage to proteins, membrane lipids, DNA, RNA. There's mitochondrial dysfunction. This series of presentations focus a lot on mitochondrial dysfunction because of the central role of mitochondria in energy metabolism, ATP production. Also, the mitochondria are the major source of oxygen-free radicals. If you remember, the oxygen that's needed for oxidative phosphorylation to generate ATP uh, is generates the free radical superoxide. Okay, so what else? In other uh, episodes in this Brain Ponderings podcast series, I talked with, for example, David Rubenstein about his work on autophagy and mitophagy. So during aging, there's impaired ability of cells to remove damaged molecules and organelles by targeting them to the lysosomes. So molecular and organellar waste disposal is impaired. Impaired DNA repair. In fact, there's these very rare, what are called progerias, premature aging syndromes that are caused by mutations in DNA repair enzymes. Werner syndrome, for example, or Bloom. And so the point there is that impaired DNA repair is sufficient to cause aging. In neurons, there's impaired ability of neural networks to maintain excitability and activity within normal limits. And in particular, there's hyperexcitability that occurs as a result of changes in glutamatergic neurons, excitatory neurons, and the GABAergic inhibitory neurons. There's evidence in the brain where there are stem cells, namely the hippocampus, that there's stem cell exhaustion of neuronal stem cells. There may also be impaired production of new glial cells from glial precursor cells. inflammation occurs in the brain during normal aging as a result of activation of microglia, which are essentially the brain macrophages, the innate arm of the immune system. So the local inflammatory response, that inflammation may occur in part as a result of damage to neurons or other processes. Something we worked all out in my lab over the number of decades is, and found evidence for impaired ability of neurons to respond adaptively to stress by, for example, upregulating antioxidant defenses to prevent oxidative damage, or stimulation of DNA repair, or stimulation of autophagy, which, as I mentioned before, fasting stimulates autophagy. Physical exercise can stimulate autophagy. And then calcium turns out to be a very important ion in brain aging and neurodegenerative disorders because, well, it plays a fundamental role in signaling and, for example, the release of neurotransmitter from presynaptic terminals, the activation of postsynaptic neurons, and the stimulation of gene expression, that is turning on genes in response to activity, neural networks. Calcium plays roles in all those normal aspects of synaptic plasticity, critical role in learning and memory, for example, and in controlling the growth of neurons and the formation of synapses. But with aging, the ability of neurons to regulate calcium properly is impaired, and that's linked closely to aberrant neural network activity. Okay, and then damage to the ends of chromosomes may occur with aging in the brain, and cell senescence, which typically occurs in mitotic cells such as glial cells, that they stop dividing, they generally get larger, physically larger, and they start spitting out a lot of inflammatory cytokines, things like tumor necrosis factor and interleukin-1 beta. So this cell senescence is increasingly recognized as playing an important role in aging in general, and recent work, including some from my lab, suggests there may be a role for excessive cell senescence in neurodegenerative disorders like Alzheimer's. Okay, so I also wanted to point out that there's problems often with the blood flow in the brain during normal aging, problems with the blood vessels, obviously in stroke. It's a major problem and an acute problem of clot forming in the blood vessel, usually as the result of age-related atherosclerosis, and diet, exercise, or lack thereof, related atherosclerosis. Right, and then you have neurons in the other cells, the glial cells. Now, one thing that we know pretty much for sure is that synapses are particularly vulnerable to aging because of their high levels of stress caused by ion fluxes and the need to restore the ion gradient across the membrane. So there's ionic stress, and there's also oxidative stress, because when the synapses are active, there's increased calcium influx into the cell, calcium uptake by mitochondria, and that calcium stimulates increased oxidative phosphorylation and formation of free radicals. So in the synapse, this is a presynaptic terminal with the neurotransmitter containing vesicles. This is a postsynaptic dendrite. There's mitochondria in these compartments. And so there's a lot of ionic and oxidative stress occurring there. So this diagram is kind of a summary of the current thinking on what happens during normal aging that may explain in part, or in large part, the changes in cognition, for example, reduction in synapse numbers or maybe some mild inflammation that occurs in the brain during normal aging. And when these age-related changes are exacerbated in conjunction with disease-specific changes, that can lead to diseases. So, for example, in cells, we know impaired mitochondrial function, oxidative damage, these things I talked about in the previous slide. Membrane lipid peroxidation, very important. Generation of free radicals in membranes, which cause the release of kind of these toxic lipid bits or cleavage products, if you will. Ionic stress, excitotoxicity, so damage to neurons caused by overexcitation and calcium influx, paired autophagy repair. Okay. So these age-related changes can promote disease processes. They can promote the accumulation of the amyloid beta peptide in the plaques in Alzheimer's disease. They can promote the self-aggregation and accumulation of the microtubule-associated protein tau in neurons, in Alzheimer's or in frontotemporal lobe dementia, for example. In ALS, Lou Gehrig's disease, also in frontotemporal dementia, and even in Alzheimer's disease or other dementias, this TDP-43 accumulates and doesn't carry out its normal function. Alpha-synuclein in Parkinson's accumulates in neurons and is thought to contribute to the damage. So you get these aggregating proteins accumulating in neurons somewhat during normal aging, much more so in the diseases. And in turn, as these proteins aggregate, it compromises or exacerbates these age-related changes. So you get this vicious cycling between the age-related changes and the disease-related protein pathologies. Also, as I mentioned in the previous slide, the ability of cells to respond adaptively to stress is impaired during normal aging, and that's exacerbated in the diseases. For example, there are neurotrophic factors, proteins in the brain that normally function to promote the growth, formation of new synapses, that they play important roles in learning and memory and other types of synaptic plasticity. They're important for protecting against mood disorders, or well, particularly anxiety and depression. And so these are compromised somewhat during aging and much more so in the disease. So as I mentioned before, the synapses are sites that are most vulnerable to these changes. And when these start to degenerate, you get cognitive impairment, or dementia, Parkinson's disease, or ALS. This is just examples of what we think might be going on in Alzheimer's, Parkinson's, and stroke. And this is just another way of looking at what I've already talked about. So in this case, I have at the things upstream of the Alzheimer's pathology, that the plaque, A beta plaque deposition, and the tau pathology, neurofibrillary tangles in the neurons. So for example, mitochondrial dysfunction, contributing to oxidative stress, dysregulated calcium handling, accumulation of dysfunctional mitochondria, impaired autophagy or mitophagy, and then you start to get increased production of the amyloid beta peptide from its precursor protein. And then as that accumulates, you get and tau pathology. These calcium handling and oxidative stress probably is involved in the tau pathology. And then downstream of these, you get these changes that lead to neural death. In the case of alpha-synuclein, a lot of focus is, in Parkinson's, most of the focus has been on alpha-synuclein, but it turns out that alpha-synuclein pathology is downstream of age-related changes or genetic factors, which I'll talk about, I think, in the next few slides. There are certain gene mutations that can cause either Alzheimer's disease or Parkinson's disease, and those mutations impact on these normal age-related processes. In stroke, you get acute deprivation of energy to neurons. So we call it oxygen glucose deprivation. And that, in the area most affected, if the blood supply is completely shut off to part of a brain region, we call it the core region of the damage following a stroke. Neurons die, all the neurons die in that region. Then there's a surrounding region that we call the penumbra, where some neurons die, some may be damaged but not die, and this is the brain region area, this penumbral region around the core that we try to keep alive in our experimental models and in translational research in clinical trials. But anyway, you get clot formation, reduced oxygen and glucose. You get bioenergetic failure, calcium overload, neural death, or damage. All right, so most cases of Alzheimer's and Parkinson's diseases, there's no known genetic cause. For Alzheimer's, there is a risk factor, the apolipoprotein E4 allele increases risk. But there are, as I mentioned, and usually the onset of the disease, the symptoms first appear in most cases of Alzheimer's or Parkinson's when the individual's in their 70s, 80s. In rare cases caused by gene mutations, the disease can manifest as early as in the 30s, typically in the 40s or 50s. In the case of Alzheimer's disease, there are two genes essentially that have been identified to cause early onset Alzheimer's. One is the amyloid precursor protein, the source of the amyloid beta peptide that forms the plaques. The other is presenilin 1, which is membrane, well, they're both membrane proteins, APP and presenilin 1. Presenilin 1 is an enzyme that one of its functions is to cleave the amyloid precursor protein and release the amyloid beta peptide, although it has other functions, and these mutations have other effects besides just affecting the amount of amyloid generated. Then in Parkinson's disease, there's about a dozen different genes that have been identified in which mutations cause early onset, cause early onset Parkinson's disease. And I'll talk about a few of those in a minute. Huntington's disease is purely genetic, caused by mutations in the huntingtin gene, and that causes this abnormal polyglutamine repeat. So the amino acid glutamine is added to the huntingtin protein essentially in a chain, and that causes the disease. ALS, mutations in superoxide dismutase 2 can cause disease, as well as at least a couple other genes. Okay, before I move on, I wanted to mention one thing that has been of interest to me, and that is that when these genes start to be identified that cause the disease, particularly those that are inherited in a dominant manner, such that if your mother or father has the mutant gene, you have a 50% chance of developing the disease. And if you get the gene, you have a 100% chance. So it is possible to eliminate those genes from families in one generation. And I wrote an article on this, published it a few years ago. And the way this is done is, for example, if you were, say, a teenager and one of your parents uh, was symptomatic with Parkinson's or Alzheimer's, they could have their DNA sequence, see if they have a gene mutation. If they do, then you can get your DNA sequenced. And if you have the mutation and you still want to have children, but don't want them to have a 50% chance of having the disease, what you can do is something called in vitro fertilization and embryo selection, where essentially you use the same IVF technology to fertilize eggs. So the father's sperm, say you're the father that has the mutation, uh, you do in vitro fertilization, your sperm, the mom's egg, and then this is done in a culture dish essentially. And then what happens if uh, when the egg starts dividing, you can, for the first few cell divisions, you can actually take one of the cells and genotype it to see if it has the mutation. If it does, then you don't use that developing embryo to implant into the mom's uterus. What you want to do is find fertilized egg in the first stage, few cell division stage embryo that doesn't have the mutation. That would be implanted into the mom's uterus, and that child will not have the mutant gene that caused this devastating disease. Of course, another way would be if you find you have the gene mutation, you can decide not to have kids or adopt children. Okay, quickly, I mentioned the tau pathology, the amyloid accumulation in Alzheimer's disease, the alpha-synuclein in Parkinson's, and the TDP-43 in ALS, and also in frontotemporal dementia, like Bruce Willis, the Die Hard star actor, has now. Uh, this diagram just shows that the disease process in these different brain disorders moves transneuronally, it's thought, through neural circuits. So, for example, in Alzheimer's disease, the first brain regions that are affected with the tau pathology and amyloid as well as are in the temporal lobe, the hippocampus, amygdala, and those kind of entorhinal cortex. And those are brain regions critical for short-term learning and memory, are also in emotional behaviors. In Parkinson's, the pathology actually in the brain is first seen in the brainstem, and it propagates upwards into the dopaminergic neurons in the midbrain and then beyond up into the cerebral cortex. And often Parkinson's patients will, as the disease progresses, develop cognitive impairment or behavioral problems controlling mood that are related to pathology in regions other than the substantia nigra dopaminergic neurons. And then in ALS and FTD, the TDP-43 pathology, in ALS, it may start in the upper motor neurons or lower motor neurons. All right, so in Alzheimer's disease, on the right here is just shown in black, stained with a silver stain, actually, neurofibrillary tangles, and then the yellow is actually diffuse and plaque amyloid beta peptide, and very often you see associated with the amyloid degenerating neurites, dendrites, neurons. Now, it's been known for a long time that the ability of neurons in the brain to take up glucose from the blood is and use it to produce energy is compromised in Alzheimer's disease. This is just an example showing PET imaging where radiolabeled 2-deoxyglucose is injected into the blood of the individual, circulates to the brain, and then this 2-deoxyglucose will accumulate in neurons because it's not metabolized in contrast to normal glucose, which is broken down to use uh, for uh, ATP production ultimately. So anyway, reduce glucose utilization in AD. Okay, so let me pause a minute. Hello? Yeah, no, he's gone. All right, bye. Now we think that the amyloid beta peptide, as it's aggregating, contributes to this impaired glucose transport in the neurons in the brain. And we provided evidence for this a long time ago in these articles I cite here. Had a graduate students, Bob Mark and Jeff Keller, who did this work. And what they found essentially is that, I'll come back to that. Uh, so the amyloid beta peptide shown in red here, this peptide fragment of the amyloid precursor protein, APP. APP is a transmembrane protein, and it's the amyloid beta peptide is generated by two enzymatic cleavages, one by beta secretase or base at the N terminus of A beta, and the other by gamma secretase, the C terminus. And then that amyloid beta peptide can self-aggregate, particularly in the presence of uh, conditions that occur during normal aging, like increased oxidizing environment. Okay, there's also another enzyme that cleaves the amyloid precursor protein in the middle of the A beta sequence, and that releases a secreted form of APP, which we and others have evidence plays a role in normal function in modifying synaptic plasticity and controlling neural excitability. In fact, we had a I had a postdoc, Katsutoshi Furukawa, read a Nature paper in 1996 where he provided evidence that this secreted APP alpha uh, reduces excitability of neurons and protects neurons against excitotoxicity. Okay, the amyloid beta peptide, as it's aggregating, particularly in the presence of low levels of ionic iron or copper, can cause oxidative stress in the membrane, lipid peroxidation, and generate a lipid peroxidation product called 4-hydroxynonenal, has 9 carbons. So anyway, that's released when these free radicals attack the double bonds in unsaturated lipids in the membrane. And interestingly, this H&E, it's an aldehyde that can covalently modify, that is, bind essentially permanently to proteins on cysteine, lysine, and histidine amino acids. And we found that this aldehyde generated in response to amyloid beta peptide and lipid peroxidation can impair glucose transporters in neuron. It can impair the function of the ion pumps that pump sodium and calcium back out of the cell after the neuron fires, and you get membrane depolarization and sodium and calcium influx. So in this way, membrane oxidative stress in AD may compromise the ability of neurons to acquire energy source, glucose, and compromise their ability to maintain ion homeostasis and promote excitotoxicity. And we think the accumulation of the amyloid beta peptide is occurring mostly at synapses. And the reason is pretty simple, because the amyloid precursor protein is transported. It's synthesized, we think, in the cell body, or most of it, and it's transported along microtubules out to the presynaptic terminals, where it is inserted into the membrane. And so the amyloid beta peptide then is generated at synapses, and this is probably a reason that it's so potent in making neurons vulnerable to hyperexcitability and excitotoxic damage to dendrites, for example, which degenerate very early in Alzheimer's. Parkinson's disease. Alpha-synuclein accumulates inside neurons in Parkinson's disease. Sometimes when it get really large, dense spherical accumulations, thousands of molecules of alpha-synuclein, and those are called Lewy bodies. But you can also get, lower levels of accumulation of alpha-synuclein that are probably contributing to dysfunction of synapses and cell degeneration. Now, normally cells remove alpha-synuclein by a process called ubiquitination. So a small protein called ubiquitin is added to the alpha-synuclein, and that's affected by enzymes, ligases, so ubiquitin ligases that ligate the ubiquitin to the alpha-synuclein. And this is just shown here. And this FYI, the ubiquitin is added to lysine residue. So lysine is a very important amino acid for all sorts of functions in cells. I mentioned before, in the context of sirtuins like SIRT3, lysines can be acetylated and deacetylated. They can be, as I mentioned, modified by this lipid peroxidation product, which actually impairs the ability of those lysine amino acids to be acetylated or ubiquitinated. Okay, when alpha-synuclein is ubiquitinated, that targets the alpha-synuclein for degradation in a large protein complex called the proteasome. Okay. So in Parkinson's, there's impaired proteasome activity, and that probably contributes to the accumulation of alpha-synuclein. On the right here are shown in black 3 different proteins. Well, 4 different proteins, including alpha-synuclein. Now, mutations in each of the genes that encode each of these 4 proteins can cause Parkinson's disease. In the case of alpha-synuclein, the mutations increase its accumulation and reduce its ability to be removed by ubiquitination. Parkin and this UCHL1, these similarly seem to be involved in normally in removing alpha-synuclein. In the case of Parkin, it also seems to be involved in recognizing dysfunctional mitochondria and targeting them for mitophagy. And PINK1 also seems to be important in regulating mitochondrial function and quality control. This just looks at what was on the previous slide in a little different way, with alpha-synuclein here forming aggregates, and shown here are different genes that are associated with these different processes in the neuron. So protein and membrane trafficking. LRRK2, mutations in that can cause Parkinson's disease. These other proteins are also involved in the same process that this LRK2 is involved in. I mentioned the ubiquitin protein system, Parkin, Parkin here, another one, DJ1, seems to be involved in reducing oxidative stress in the mitochondria, and so on. So there's a lot known about the pathogenesis of Parkinson's disease at the cellular level because of the identification by geneticists of these mutant genes in families that have inherited Parkinson's disease. But there's also evidence in the case of Parkinson's disease that certain environmental toxins may contribute to some cases of Parkinson's disease. Now, there's a very interesting story, kind of like a mystery story, murder mystery story, if you will, that took place in California. Bill Langston, neurologist at a clinic there in Santa Clara, California. One day, one of his residents, neurology residents, who he's training, came to him and said, I just saw a patient that, based on my evaluation, has all the symptoms of Parkinson's disease. However, this patient is young. He's only in his early 30s. And moreover, he doesn't have any history of development of tremors and motor dysfunction that symptoms seem to appear within a few days. And then over the next few weeks, several other patients came in with the exact same Parkinson-like problem of acute onset with no preceding history. Okay, so that was very bizarre. And Bill Langston, he tried to figure out what the heck was going on. And in taking the history of these patients, they all were drug users, they all were heroin users. And then with the help of the law enforcement in the area, Langston was able to find out that all of the affected individuals that came into his clinic had gotten their heroin supply from the same source of synthetic batch of heroin. It turned out then they got hold of some of that batch of heroin that was confiscated by the police, and they analyzed it and found that it had high levels of a chemical called MPTP. So it was they screwed up. The people who were making the heroin screwed up, is the bottom line. And it had this MPTP in it. And then they found if they give this MPTP to mice or rats, and later on studies were done in monkeys, that it will cause the degeneration selectively of dopamine neurons in the substantia nigra and will cause motor dysfunction, kind of like Parkinson's symptoms in humans. But then, however, cell culture experiments, if they exposed neurons directly to MPTP, it didn't seem to be real toxic. So the additional work showed that what happens is this MPTP gets into the brain pretty easily. It gets into glial cells, astrocytes, and there's an enzyme called monoamine oxidase B, which is normally functioning and metabolizing neurotransmitters. And they found that this enzyme converts MPTP to MPP plus, so it's oxidized essentially. And this MPP plus then is recognized by the dopamine transporter that normally transports dopamine into the dopaminergic neurons after it's released at the synapse. And so this MPP selectively accumulates in dopamine producing neurons. And then it turns out it's a very potent inhibitor of mitochondrial complex 1 and the electron transport genes. So it causes energy failure selectively in the dopaminergic neurons. It also causes oxidative stress. So that's why the neurons die. There are other environmental toxins, for example, rotenone, pesticide, that also inhibits mitochondrial complex 1. And if rotenone is given to rats or mice in high concentrations, or even in moderate concentrations over long time periods, it can lead to degeneration of dopaminergic neurons and other neurons. And there's some evidence epidemiologically that individuals who work in the agricultural industry, for example, immigrant farmers, including undocumented farmers, which if we get rid of, it's going to cause a big problem in our food supply. But unfortunately, these individuals uh, are, can be exposed to chronically to certain pesticides that have the potential to increase their chance of developing a Parkinson's disease. Okay, I just want to mention that so genes in which mutations cause early onset Parkinson's disease, obviously they've been cloned, and so the amino acid sequence is known, and they have interesting functional domains and evidence is emerging for interesting normal functions of these proteins and therefore um, reasons why mutations cause disease. So, for example, alpha-synuclein uh, has a couple different domains. This one may be involved in self-aggregation, um, and the mutations are in certain regions uh, that may be involved in those processes. It's not completely clear what the normal function of alpha-synuclein is. There's some evidence it may play a role in synaptic plasticity, but more needs to be done. This LRRK2, it has very interesting domains. It has a kinase domain, so it can transfer phosphate from ATP to many different proteins in cells that are targets and affect their function in that way. And mutations tend to impair the kinase activity, but it also has, for example, this ankyrin repeat domain indicating that may be associated with the cytoskeleton and be involved in, for example, cytoskeleton membrane interactions. DJ1, PINK1, Parkin, uh, again, amino acid sequences are known and a lot of work going on to understand the normal function and roles in disease. Yeah, just another way of looking at this Parkinson story, which is really interesting and provides a lot of potential targets for therapeutic intervention by developing drugs that target, for example, alpha-synuclein accumulation or enhance its removal by the proteasome or um, modify mitochondrial dynamics, mitophagy, for example, or for example, something simple, just uh, reduce mitochondrial oxidative stress, uh, enhance ATP production, etc. Okay, one really interesting thing with Parkinson's disease, among many other, is that the disease process, the alpha-synuclein accumulation in cells begins not in the brain, but in the intestines, in the gut, and in particular in neurons in the parasympathetic nervous system, that is, axons from in the vagus nerve that innervate the gut and the neurons in the gut. Now, the vagus nerve, the cell bodies of the cholinergic neurons, neurons that use acetylcholine as their neurotransmitter, are in the brainstem. dorsal motor nucleus of the vagus. So the cell bodies of these vagal parasympathetic neurons are in the brainstem, and their axons course through the vagus nerve to the heart, shown here, or the gut, which I guess I should have shown here. Um, the parasympathetic nervous system slows heart rate and increases heart rate variability, which is a good thing. And that happens in people who do a lot of aerobic exercise, or we found even with intermittent fasting will enhance this parasympathetic tone. And that kind of counteracts the sympathetic nervous system, which is the flight or fight stress response system. In the case of the gut, the parasympathetic nervous system enhances gut motility. And it's been known for a long time that patients with Parkinson's disease often experience chronic constipation. So that's consistent with, impaired parasympathetic activity. And then Heiko Brack, along quite 20 years ago, provided evidence by immunostaining with alpha-synuclein antibody that in the brain, alpha-synuclein pathology is first seen in early stages of Parkinson's in the brainstem and then progresses up to the midbrain dopaminergic neurons and other brain regions. And then because he saw a lot of alpha-synuclein in the dorsal motor nucleus of the vagus neurons in the brainstem, they looked at the gut target of these axons and found alpha-synuclein accumulating there. And then there's evidence, clinical evidence actually, that people who receive vagotomy for intestinal problems, for example, well, mainly very severe intestinal ulcers, that if they cut the vagus nerve, then they follow these people over decades and decades, they don't develop Parkinson's disease. So if you remove the vagus innervation of the gut, that seems to protect in humans against Parkinson's. And then studies in animals confirm this. That is, that if you cut the vagus nerve in transgenic mice, for example, that overexpress mutant self-aggregating alpha-synuclein, then they don't develop the brain pathology or motor dysfunction. And there's a lot of work now going on to understand what's going on in the gut that causes the initial alpha-synuclein pathology that progresses transneuronally through the vagus up to the brain. One thing that seems to be emerging is chronic gut inflammation may play a role. And there's a number of disorders that, where there's chronic gut inflammation. I had a postdoc, Yuki Kishimoto, who found that in the alpha-synuclein mice, so same mice used here essentially in this study, that if when he induced chronic mild gut inflammation, there's a way to do that by administering dextran sodium sulfate. The animals causes mild gut inflammation, which we documented. And then we found that that caused exacerbated alpha-synuclein accumulation in neurons innervating the gut and in the brain. So there may chronic gut inflammation can, at least in the mouse model, exacerbate the disease process. Now, in ALS, I'm not going to spend much time at all on ALS, but in ALS, the upper motor neurons, the neurons in your motor cortex that when they fire, initiate movements of your muscles consciously or unconsciously, and then they synapse on the lower motor neurons in your spinal cord, and their axons then project to muscles in your arms, legs, wherever, cause contraction. There's a lot of evidence that oxidative stress, ionic stress, excitotoxicity play a role in the demise of motor neurons in ALS. And Steve Rothstein, colleague at Hopkins, in fact, developed the only drug that has any effect on the progression of ALS. It slows the progression statistically significantly, but not dramatically. And that drug, uh, essentially the bottom line of what it does is it reduces the amount of glutamate present at synapses and thereby reduces excitotoxic damage, we think. There are a number of genes that have been associated with ALS and FTD. I mentioned TDP43. There's also this fuse, actually SOD1 isn't shown here. So anyway, there's a lot of genetics there. Okay, now in the second half or maybe last third of my presentation, I'm going to talk about some things I talked about in the previous presentation, but I'm going to then quickly shift to pharmacological interventions that target specific changes that occur in cells during aging, that target dysregulated energy metabolism or mitochondrial dysfunction or autophagy or uh, cell senescence, for example. Okay, so I talked about how exercise and intermittent fasting, intellectual challenges may protect neurons against age-related diseases. Previous presentation. Uh, it's also known how these age-related changes may exacerbate this, and particularly the age-related changes can be exacerbated by a sedentary, overindulgent lifestyle. So a chronic positive energy balance, if you will, or another way to look at that is a lack of bioenergetic challenges of exercise or intermittent fasting, caloric restriction. So we know that obesity is, well, it's now designated as a disease, but it often involves insulin resistance, including in neurons in the brain, synaptic dysfunction. There's also good evidence from animal studies that diabetes and obesity can reduce levels of neurotrophic factors, such as brain-derived neurotrophic factor in the brain. So you get impaired adaptive stress responses. If you load rats or mice up with, you put fructose in their drinking water and you give them a high saturated fat diet, if you do that lifelong, they die early. But in the brain, this obesogenic diet, if you will, causes oxidative stress, impaired autophagy, and renders neurons vulnerable to excitotoxicity. That's been shown by many labs, including ours. And in humans, there's good evidence that chronic obesity or type 2 diabetes accelerates age-related structural and functional um, aspects of the brain, including cognition. Even there's evidence that adolescents with obesity, on average, perform more poorly in cognitive tests than compared to age-matched, healthy body weight uh, adolescents. Okay, back in the 1990s, we showed using neurotoxin models of Parkinson's, Alzheimer's, and Huntington's disease that every other day fasting, intermittent fasting, can protect neurons against dysfunction and degeneration. We also found that when we have animals, mice or rats, on intermittent fasting before we do an experimental stroke, where we transiently shut off the blood supply to a particular blood vessel in the brain, that sort of pre-treatment, if you will, with intermittent fasting results in less brain damage and less uh, dysfunction of the brain, less symptoms. And these are just examples from some of those studies. This is the first study Anna Bruce Keller did, where in a model of epilepsy, where she induces epilepsy with a neurotoxin that activates glutamate receptors excessively. So the black here is neuronal cell bodies in the hippocampus. So you see here in this area called CA3, there's not much black. That means a lot of neurons have died with this dose of this excitotoxin in the animals fed ad libitum. In the animals that were subjected to food restriction, every other day fasting, the toxin caused very little or no damage at this dose. So there's protection against excitotoxicity. Similarly, in the stroke model, there's less less brain damage and less motor dysfunction when the animals, in this case rats, are maintained on intermittent fasting prior to the stroke. In the MPTP model of Parkinson's disease, actually, Wenjian Zhuan, who's a full professor at Hopkins now, when she was a postdoc in my lab 20 years ago, showed that intermittent fasting can protect dopaminergic neurons against MPTP toxicity. And then we followed up that soon after that with in a monkey model, rhesus monkey model, where we selectively damage dopaminergic neurons on just one side of the brain of the monkeys, and they have problems with controlling their hand movements on the other side of the body, because one side of the body controls movement on the other side of the one part of the brain controls movement on the other side of the body. But anyway, the bottom line is, in multiple models, intermittent fasting was protective, and we, as I talked about in the previous episode of Brain Ponderings, have looked into what's happening in neurons that explain this. But there's a lot going on that intermittent fasting does, and essentially everything we've looked at in terms of the hallmarks of brain aging, intermittent fasting seems to counteract those age and disease-related changes. We found that daily time-restricted eating with a 30% reduction in daily calorie intake, as well as alternate day fasting, reduce brain damage and functional outcome in a mouse model of Alzheimer's disease that we developed with Frank LaFurla out at UC Irvine. These animals get amyloid accumulation, shown here. The green is amyloid plaques outside of neurons in the cerebral cortex and hippocampus, and on the right is hyperphosphorylated tau, the kind of tau that self-aggregates, and you see accumulation of the tau in neurons. It's a slow process in this mice, slow for mice. This is after a year and a half, and we don't see much pathology until the animals are getting towards the 6 months to a year. We start to see pathology, and then it accelerates. And when we test these animals with the different diets, we start them at 5 months of age on either ad libitum, every other day fasting, or daily time restricted eating with calorie restriction. And then we had non-Alzheimer's mice fed ad libitum, and we let them go for a year. And then after a year, we tested them in the water maze, and you can see that the learning curve in the Alzheimer's mice fed ad libitum is not very steep. They don't quickly learn to solve the maze, whereas the intermittent fasting protected against that. Interestingly, though, the intermittent fasting didn't have a real dramatic effect on the amyloid pathology, suggesting that maybe the intermittent fasting is actually protecting the neurons against the accumulating amyloid, as opposed to, or in addition to, reducing amyloid accumulation. Right, this is some data from the mouse, the monkey MPTP model. This is a rhesus monkey. Uh, and so what we did is, in this study, we had the animals on 30% daily calorie restriction. This is their body weights, so the calorie restricted animals are lower. So we let them go for 6 months on either the calorie restriction or normal feeding regimen. And then we looked at dopamine levels in their brain using PET scanning with a radio tracer. And then we injected MPTP into the one carotid arteries, damages dopaminergic neurons on that side of the brain, causes motor impairment on the contralateral. And then we have a neat way of testing the motor function. Um, Monkeys like lifesavers, these candies with a hole in the middle. And you can put these candies on a wire, a straight wire, for example, or a curved wire. And on the straight wire, they can pretty easily get the lifesaver off and then eat it. On the curved wire, where you got to twist the lifesaver, if their dopaminergic system is compromised, they have a lot of problems. And so that's this map testing. And then we did MPTP, post-MPTP PET imaging and motor function testing, and we found that the caloric restriction lessened the impairment in motor function, two different ways of measuring this fine motor function. And there was significant preservation of dopamine levels in at least two parts of the striatum, which is where the axons of the dopaminergic neurons protect. And there was also the caloric restriction resulted in higher levels of the two different neurotrophic factors, GDNF and BDNF, which were previously shown in experimental models to be able to protect neurons, dopaminergic neurons, in models of Parkinson's. Okay. Also, very recently, Dimitros Kapagiannis, a neurologist who was with me and is now a tenured investigator at National Institute on Aging, where I was his lab chief. Dimitros, together with me, designed a study where we took people between the ages of 55 and 70 with obesity and insulin resistance. So these are people at risk for cognitive impairment and Alzheimer's disease. We randomly assigned them to 5-2 intermittent fasting, 2 days a week, eating only 480 calories a day, the other 5 days eating normally, or the USDA healthy living diet, which is just counseling on what to eat and how much. And then before we started them on these diets, and 2 months later, we did a lot of measurements, body weight, blood insulin, glucose levels, a lot of cognitive testing for memory, executive function, and so on. And then we have some ways to look at brain insulin resistance and other markers in the cerebrospinal fluid, for example. And we found that with both diets, the subjects lost weight. So this is, for example, sorry, this is baseline, week 4, week 8. So there's modest but significant reduction in body weight and waist circumference. Insulin levels are down significantly more so with intermittent fasting. And this is also true with this indicator of insulin resistance. So the intermittent fasting has a very good beneficial effect in improving glucose regulation, reducing glycated hemoglobin, for example. The ketones in the blood go up, particularly with the intermittent fasting, somewhat with advice for healthy eating, probably because these people are eating a little less because of the advice, hopefully. So the ketones go up. cholesterol goes down, more so with the intermittent fasting. And then for cognition, in measures of executive function, which is essentially it's your ability to carry out cognitively challenging tasks. And there's different ways to look at this. For example, this set shifting test looks at your ability to change your focus, and you're first working on this and then switch your attention to this and quickly get on task. Fluency is this kind of efficiency in processing information. So these improved and significantly more with the intermittent fasting. In fact, with the intermittent fasting, there were statistically significant improvements in cognition in both executive function, shown at the top here, and in memory. Essentially, for example, being able to recall a sequence of words that you were exposed to 10 minutes earlier, a half hour earlier. There were no major effects on amyloid in the cerebrospinal fluid or tau. We did look at functional, both structural and functional MRI. And for the structural MRI, we actually had some evidence for a kind of slowing of age-related structural changes in the brain. Okay, what about pharmacological interventions? Ketone esters, I mentioned this before. There's a lot of animal studies showing that if you supplement the diet of rats or mice with the ketone ester, beta hydroxybutyrate, it is neuroprotective in models of Alzheimer's, Parkinson's disease. Another approach to bolster cellular bioenergetics is to give nicotinamide riboside, which will boost NAD levels in neurons, which, as I mentioned in the first presentation in this series, well, NAD is very important for the function of enzymes like sirtuins and others, and in energy metabolism. Another approach actually is to pharmacologically impose a mild bioenergetic stress or other type of stress on neurons. And the idea intermittently. So the idea is to kind of mimic the effects of fasting or exercise or normal activity in neurons like occurs during learning and memory. And so the idea is to engage adaptive stress responses in the neurons. For example, one way is this mitochondrial uncoupler, uh, 2,4-dinitrophenol, or its long-acting compound MP201. And this has been shown in animals to induce stress responses in neurons, including increased production of brain-derived neurotrophic factor, increased levels of protein chaperones, and also, interestingly, it can affect the mTOR pathway, inhibit it, which is what's seen with fasting and exercise. Another approach is there's potassium channels in mitochondria, and this chemical diazoxide was actually developed to treat high blood pressure, hypertension, and it does. It's actually very good at high doses in reducing blood pressure. But Donglu and my lab, over a number of studies over a number of years, and others in other labs have found that diazoxide at mild levels that cause very little or no change in blood pressure can induce adaptive stress responses and be neuroprotective in models of Alzheimer's or stroke. Another type of stress one can do is supplement the diet of animals with 2-deoxyglucose. So you're causing calorie restriction at the cellular level. It induces stress responses, and it's neuroprotective. Rapamycin, this mTOR inhibitor, a lot of work on that. showing benefit in animal models of neurodegenerative disorders, and there's clinical trials in humans. With that, there's actually clinical trials in humans with DNP and ALS and with nicotinamide riboside and ketone ester in Alzheimer's disease. Another approach is GLP-1 receptor agonists that you've heard a lot about in the news lately, based on the trade names, the company names, Ozempic, for example, Wegovy. Um, we did some work together with Josephine Egan and Nigel Gregg at NIA with Xendin-4, GLP-1 receptor agonist, metformin, and these drugs are used to treat diabetes. These two, and there's some work there. Okay, a lot of approaches. As I mentioned, we think that in Alzheimer's, at least in the early stages, while neurons may not be able to take up and use glucose, they can still use ketones. That was shown by Steve Cunan in his work. And we found in an animal study that when we gave ketone ester in the diet of the 3-XTG AD mouse model of Alzheimer's disease, it lessened the cognitive impairment and lessened the the tau neuropathology. And there's clinical trials going on now with the ketone ester, including one by Dr. Kapagiannis at the National Institute on Aging. Okay, so here's just some examples of publications. This is our very early work in animal models showing that if we give them 2-deoxyglucose, it can protect in neurotoxin models, for Parkinson's in a stroke model. And then, interestingly, we also found that 2-deoxyglucose is beneficial in cardiovascular responses to stress. It can protect the heart against psychological stress. Yeah, and then in Alzheimer's disease model, Robbie Brinton's lab showed beneficial effects of 2-deoxyglucose, as well as Raj Ratan, who I did a podcast with on his work before. Here's some of our early work with the DNP, the mitochondrial uncoupler, and these models here. And then this is the diazoxide. So a lot of approaches going on. The GLP-1 receptor agonists, they're used for diabetes because, so GLP-1 is a hormone. It's produced in cells lining your gut. And it's when you eat, particularly carbohydrates and glucose, GLP-1 is released from those gut cells into the blood. It does several things that improve glucose regulation, that is, promote the removal of glucose from the blood. First, it increases insulin release from the pancreas. So more insulin, more stimulation of insulin receptors, which then stimulates the cells to remove glucose from the blood, muscle, liver cells, neurons. GLP-1 also increases the sensitivity of cells to insulin, such that with a given amount of insulin, in the presence of GLP-1, the insulin is more effective in stimulating those cells to take up glucose. But GLP-1 also acts in your brain and the hypothalamus to reduce appetite, to make you less hungry. So it's kind of a stop eating signal. That's in addition to leptin, which is another one. Okay, so numerous studies now showing that GLP-1 receptor agonists, such as exenatide, which we worked on, or liraglutide, or Ozempic, all of them, in this case, liraglutide, reduce body weight. And this is mainly because the person is less hungry. And this is interesting too, because people with obesity often have leptin resistance, which means that the stop eating signal that they normally get from leptin is compromised. So people with obesities often perceive that they're hungry even when they're obviously uh, far from approaching a starvation condition. Um, this is a study done uh, uh, by Nigel Gregg in collaboration with a group across the National Institute on Drug Abuse, our neighbors here in Baltimore. And we helped a little bit with that. And this study showed that in the MPTP model of Parkinson's disease, the GLP-1 receptor agonist exenatide protected dopaminergic neurons and lessened the motor dysfunction. So they developed less motor symptoms. So there's a protective effect in a mouse model. And then a group in England, uh, Tim Faltini, who I did a podcast with on his work on this GLP-1 receptor agonist in Parkinson's patients. And he showed in an initial study that when these Parkinson's patients are given this GLP-1 receptor agonist, uh, it, it, it not only completely prevents the progression of the disease process, but it actually improves their symptoms somewhat over a period of 60 weeks, as shown here. So this is in this Lancet paper there. My understanding is they are done with, or soon will be done with, and report on a subsequent large phase 3 study. Uh, so we look forward to seeing those results. Then this is my last slide. One thing I didn't mention really at all is cell senescence. And I actually left it out of this Hallmarks of Brain Aging article, largely discussion of it, which we published only 6 years ago, because in the last really 5 years, there's been many labs that have provided evidence that cell senescence occurs in the brain during normal aging and that it's exacerbated in Alzheimer's disease and possibly other age-related neurodegenerative disorders. And importantly, that eliminating senescent cells can have a therapeutic effect in animal models of Parkinson's disease. So I just showed 3 articles here. This is Darren Baker's had this Nature paper in 2018, and I had Darren on a podcast on cell senescence in the brain. And they showed that um, cell senescence does occur in the brain. When cells undergo senescence, uh, in dividing cells, they stop dividing, they get bigger in size, and they start secreting what we call pro-inflammatory cytokines like TNF, interleukin 1 beta. And he showed that there are senescent cells in the brain in a mouse model of tau pathology that's relevant to both Alzheimer's and frontotemporal dementia. In his study, his evidence suggested that those senescent cells come from microglia or astrocytes. Then, not long after that, Pei-Su Zhang in my lab in the 3x TGAD model of Alzheimer's disease, she found that there's cells with senescence-like phenotype secreting pro-inflammatory cytokines that accumulate more in the brain in these Alzheimer's mice compared to control mice, and that these senescent cells are typically associated with amyloid deposits. And then when she tried to identify, are these microglia, astrocytes, or another cell type, her evidence suggests that they're actually oligodendrocyte progenitor cells, the stem cells that give rise to the myelinating uh, oligodendrocytes, the cells that wrap around and insulate the axons of neurons. And that's interesting because throughout the adult brain, uh, the oligodendrocyte progenitor cells are present and they're capable of dividing, and they're capable of differentiating into oligodendrocytes and myelinating cells, for example, if there's damage to an axon. And she found that when she administers what are called senolytics, which is a drug cocktail of a natural product called quercetin and a drug used to treat some cases of cancer called dasatinib, that eliminated many of the senescent cells and ameliorated the cognitive deficits. So that's shown here that this senolytic treatment targeting the senescent cells, the animals, this is the control animals, Alzheimer's animals. This is with the senolytic dasatinib plus quercetin. They have a steeper learning curve. And when you then wait a few days and test their memory, they're better able to retain the memory when the senescent cells are removed. Okay, so this is the last in the four-part series of brain bioenergetics. I hope you got a lot of information from this. You can, if you want, you can email me or in YouTube or YouTube for sure. You can in the comment section. Feel free to ask me questions. I can send if you want any of the articles I cited in these slides. I'm happy to send them to you by email. It's Thanksgiving's coming up this coming week, and actually this week, and next week I may or not have a podcast, depending on the schedule. I'm still trying to get one particular investigator to do a podcast with me for that week. So have a good Thanksgiving. Bye.